Greetings, Biology 112 students. Here is a recording where we summarize the highlights and ideas of the urinary system. This is going to relate to chapter 24 in the McKinley textbook. And we see that the urinary system is a system whose main job is filtration of blood and elimination of the wastes. Let's back up a little bit and think about some of the systems we talked about over the last semester. We talked about the cardiovascular system with its blood. We talked about how the respiratory system puts gases into that blood and gases out of that blood. We talked about how the digestive system puts nutrients into that blood. And here we look at how that blood gets filtered. So the main functional unit of the urinary system is the kidneys. And so in this series of slides, we're going to look at the external and internal anatomy of the kidneys. We're going to look at the blood flow through kidneys and nerves. And we are going to see how nephrons are the functional unit of the kidney. So the idea is that the kidneys filter fluid from the blood. We're going to call this filtrate. And we're going to need some substances to stay back in that bloodstream, and that's called reabsorption, whereas we get rid of wastes, toxins, and excess ions through a process called secretion and excretion. So we start off talking about an overview, just some gross anatomy of this abdominal cavity. We see two kidneys in this image, and these are located retroperitoneal. We can also see the presence of two ureters, leading to a urinary bladder, and exiting from that urinary bladder, is going to be the urethra for both male and female structures. Again, as we said before, the function of having a urinary system is that we filter blood and return most of that water and solutes back to the bloodstream. And we can see that in this image where we have the inferior vena cava moving through, receiving blood from the kidneys. And here we have the abdominal aorta delivering blood to those kidneys. Let's take a little bit more of a discussion on the overview of the functions for these kidneys. So one function of these kidneys is that we are going to regulate blood ionic composition. That means the kidneys regulate tiny ions like sodium, potassium, calcium, and phosphate ions to important homeostatic levels in the bloodstream. And there's also a little bit of regulation of hydrogen ions and bicarbonate levels to maintain that acid-base balance. So that kind of leads a nice segue to function number 2. Yes, we are regulating blood pH with that acid-base balance. Also going to work with osmolarity. You might recall that osmolarity involves the movement of water according to a different solute's concentration gradient. And in this series, we're going to talk about also how glucose gets regulated. We talk about how the kidneys may engage in gluconeogenesis, and this is an attempt to maintain normal glucose levels and also mitigate conditions of nutrient deprivation. Another function of having kidneys in the human body is that we are going to regulate blood volume. And this regulation of blood volume can be accomplished by conserving the water or eliminating the water. So if we want to increase the volume of blood, we're going to conserve water and not release as much of it. And of course, if we have too much blood volume, we can eliminate more water out of that blood and therefore decrease the volume of blood. Related to this is item number 4, the regulation of blood pressure. And so these kidneys are able to regulate this blood pressure by controlling that fluid volume. And again, that's through urine loss. And also, we can accomplish that by releasing renin. And this is an enzyme required for angiotensin II production, and that's going to increase blood pressure. Another function for these kidneys in our human body is the release of erythropoietin. We might recall that we discussed this hormone earlier when we discussed the blood in the human body. And what basically occurs is, in response to lower blood oxygen levels, the kidneys are going to secrete this hormone called erythropoietin, also we abbreviate EPO, and this is going to stimulate the red bone marrow to increase the erythrocyte formation. And then lastly, as we mentioned before, number 6, the excretion of wastes and foreign substances such as toxins. Here we have a transverse view of the abdominal cavity, and this is possibly a better view to see that the kidneys are located retroperitoneal, and they are also wrapped in that parietal peritoneum. That is, to be more accurate, they are bounded on one side by the parietal peritoneum, and the other side is going to be bounded by perirenal fat capsule. These are going to be about 4 to 5 inches long, approximately 2 to 3 inches wide, and about 1 inch thick. And we have 3 main tissues surrounding the outside of this kidney. Number 1 is going to be our renal capsule. We'll see that in a slide or 2 down the road. And then we can see in this diagram, we have some yellow adipose that's going to cushion his kidneys. We're going to call that the adipose capsule. And then there's going to be an outer renal fascia, a sort of connective tissue that's going to cover the outer part of it. It's going to help keep those kidneys locked in to the place where they need to be, which is roughly around the body of the L2 vertebra. Here is a frontal view of the internal anatomy of the kidney. On the last slide, we left off talking about this fibrous capsule being one structure of the kidney. And this fibrous capsule is going to be composed of dense, irregular connective tissue. This is going to adhere to the external surface of the kidney. And its function is to maintain shape. It's going to do some protection from trauma, and also going to prevent us from having pathogens infect that region. Well, coming in from that fibrous capsule, we see 3 main regions to this kidney. One main region is going to be our cortex. Often in anatomy, cortex refers to a general outer region. And similarly, in anatomy, a medulla region tends to describe a more central region of the organ. Thirdly, we see that inside the innermost region is the renal pelvis. And here we're going to collect urine for transport into the ureter. And from ureter, it's off to the bladder. In a more in-depth feature, we see that the structures inside this kidney include such thing as papilla. Here's a image of a papilla here. In our root words, papilla means nipple, so perhaps you can see a nipple-like feature in there. Also, we have pyramids, so perhaps you see pyramid-like structures in there. And then we have minor calyx and major calyx. In other words, a small space to collect that urine, and it dumps into a larger space to collect that urine. So in general, the renal cortex, this outer region, and the renal pyramids as well, constitute the functional portion of the parenchyma of the kidney. And then also we see later on down the road that the important feature of the kidney is the nephron. The nephron is the functional unit of the kidney. These are not visible in this slide. You can barely see a hint of them in the vertical striations shown. But we will definitely get more into the nephron structure later on. So in the summary, this kidney has an outer capsule that protects it, 3 main regions, and other smaller features on the side that each have their own function. Let's look more in depth into what those do. So here we have the similar frontal view, but this time we have a real kidney. Again, we see that the outer region of the kidney's parenchyma is called the renal cortex, and the inner region of the kidney's parenchyma is going to be called the renal medulla. And remember that this is going to contain those pyramids, and these pyramids are going to be separated by renal columns. Those renal papilla, those are going to be sort of the medial tip of the renal pyramid. And then again, we see this drainage system that fills the renal sinus cavity. The minor and major calyces are going to empty into the renal pelvis, which then goes into the urine. And at this point, we switch gears a little bit and talk about how that kidney is supplied with blood, and we see how that kidney is supplied with nervous tissue. So this means we're jumping a section or two ahead in the McKinley text. And we start by thinking of the aorta. I'm sure we're all aware that the aorta is a major blood vessel running through the central region of the body. And so the coming off of the aorta are going to be these renal arteries. And the idea is that the aorta is going to work with the renal arteries to deliver blood to these kidneys. And then arteries branching from the renal artery are called segmental arteries. And then we see after branching from the segmental arteries, they're going to travel through the renal columns towards the corticomedullary junction. And those are going to be called interlobar arteries. So by the way, when we're saying this word corticomedullary junction, we're talking about a junction or a boundary between the cortex region and the medulla region. So as we said, that gets us to our interlobar arteries. And then again, we're looking at the red ones, the arteries branching from the interlobar arteries are going to project parallel to the base of the medullary pyramid. These are called arcuate arteries. And then the leading to cortical radiate arteries. And by the way, you can see how these radiate through the cortex, so to speak. And we then arrive at the nephron. So this dashed line here shows us entering a microscopic feature, and that means we arrive at the afferent arteriole. So each afferent arteriole enters a renal corpuscle, and then later on we see how it's going to form a glomerulus where blood is filtered and eventually exits the renal corpuscle through the efferent arteriole. We also get a little bit of interaction with a structure called the vasaracta, which we discuss later. And at this point, most of the blood has been filtered thanks to the nephrons, and we follow a very similar pathway on the way back: cortical to arcuate, arcuate to interlobar, and to renal vein. This time, however, we are dumping into the inferior vena cava. So in summary, this pathway serves to allow kidney to filter its blood. This slide has a nice summary of some ideas about, general ideas about the kidney. We see that the kidney does get a significant amount of blood from the blood vessels, receiving about 25% of resting cardiac output. And again, that's coming in through the renal arteries. We have capillary beds. Earlier we mentioned glomerular capillaries. Again, we'll take a little bit more look at those later on down the road. That's going to produce a substance called filtrate from blood. So that's going to be an early precursor to urine. These things called paratubular capillaries are going to reclaim most of that filtrate. And then later on, we look more closely at a structure called vasa recta. This is going to supply nutrients to the medulla, and it's also going to concentrate the urine. On another note, these kidneys are innervated. They do receive a nerve supply. So each kidney is innervated by the autonomic nervous system, and that's going to occur through the renal plexus. This renal plexus contains both sympathetic nerves from thoracic nerve 10 and thoracic spinal nerve 12, and it also receives parasympathetic nerves from the vagus nerves. At this point in our discussion, we take a closer look at the functional unit of the kidney, that is the nephron. So in this diagram to the right, we see that we're taking this structure that we've already discussed, the renal pyramid, which includes part of the cortex and includes part of the medulla. And so we go more in depth in a microscopic level, and we see the basic structure of a nephron. And in this nephron is a structure called the glomerulus. That's where fluid is going to be filtered. Here's a quick look at the glomerulus. And we also have a renal tubule into which the filtered fluid passes. There we go, a big picture of that. And also, that's going to be our structure, so let's look at the function of these. The function of the glomerulus is going to be to produce a filtrate, an early stage of the urine. Later on, that filtrate gets reabsorbed in tubular reabsorption. And thirdly, we have tubular secretion. Some substances are further secreted out of those membranes. So here we have an image showing the basics of the nephron. And let's look at some of these structures. We see three main structures, the renal corpuscle, the renal tubules, and the collecting ducts. I have a little bit more simplified diagram on the right. And here we see this nephron structure sort of simplified, sort of stretched out. So as we said earlier, we have the renal corpuscle. And this renal corpuscle is going to contain a structure called the glomerulus. And the glomerulus is also going to be called the Bowman's capsule. And this is going to be a sort of conglomeration of capillary loops. And this is going to be consisting of simple squamous epithelium. So this epithelial kind of creates a cup, so to speak, and this is what's going to collect the filtrate. So I like to think of the glomerulus as or the renal corpuscle as a sort of a catcher's mitt with a softball in it. Imagine how a catcher's mitt sort of envelops snugly the softball. Here we see that the capsule snugly encases the glomerulus, and in between there is a capsular space. And in that capsular space, that's where the filtrate is going to pool off to. After our renal corpuscle, we arrive at the renal tubule. Our first stop is the proximal convoluted tubule. We see why it gets its name. Proximal because it's close to the glomerulus, and convoluted because it appears convoluted in nature. From this proximal convoluted tubule, we reach a structure called the nephron loop. The nephron loop is also termed the loop of Henle. And notice we have a descending limb and then a ascending limb. After this descending and ascending limb, and by the way, we get more into the functions of those later on. We then arrive at the distal convoluted tubule. Distal because it is more distant, but we also see that is convoluted in nature as well. And that gets us to the collecting duct. And the collecting duct is going to take that urine and send that urine down into the calyces, where it then goes to our ureter. We now serve to compare and contrast two types of nephrons, cortical nephrons versus juxtamedullary nephrons. Let's get our bearings straight here again. Notice that the top portion is called the cortex, the lower portion is called the medulla. So hence this name cortical refers to most of its stuff being in the cortex. This nephron termed juxtamedullary, the word juxta means next to. So here we are next to the medulla with a little bit more of material extending into there. This slide is nice because it shows also the functions of each structure. On the next slide is a possibly more simplified version of these two types of nephrons. So generally speaking, nephrons with a relatively short nephron loop that barely penetrate the medulla and they make up the bulk of the cortex, these are going to be called cortical nephrons. Whereas nephrons with relatively long nephron loops extend deep into the medulla, these are going to be called the juxtamedullary nephrons. And the purpose of this is going to establish salt concentration gradients that allow for the regulation of urine concentration by ADH, that is antidiuretic hormone. So in summary, cortical nephrons have a short loop of Henle, and their glomerulus is further from that corticomedullary junction. And then juxtamedullary nephrons have a long loop of Henle, and the glomerulus is closer to that corticomedullary junction. Earlier, we examined blood flow to the kidneys. So at one point, you might recall we talked about the afferent arterial. The term afferent means it's going towards something. Efferent meaning going away from that structure. So we're going towards and away from the glomerulus. And so then after leaving this glomerulus, the efferent arterioles give rise to paratubular capillaries and vasa recta. This slide serves to show urine formation. So the idea is that this blood is under high pressure in the glomerulus. Thus plasma, that stuff that's part of blood, moves into the glomerular capsule, and we call this filtrate. The only thing that stays behind are some larger plasma proteins. So we're talking about this region here. As this filtrate then moves along the tubules, we call it a tubular fluid. Most of the water and many of the other molecules get reabsorbed in the bloodstream. Some substances, however, are actually going to be secreted into the tubular fluid. So once this fluid moves into the collecting duct, additional water is going to be removed, and the urine is created, and that is a concentration of wastes. So again, that's just a brief overview. We go more in depth into those processes later on. But in general, on this slide, we see that urine formation is filtrate coming out, and then processes of reabsorption and absorption occurring in the tubules, and then the tubules will then lead to a collecting duct, not shown here, which then brings that concentrated urine. Let's go a little bit more in depth into the structure of a renal corpuscle. That is, we're going to look specifically here at Bowman's capsule. So again, let's get our bearings straight that we're looking here at a nephron, and a nephron is a part of a kidney. And that means we're going more in depth into the structure called the renal corpuscle, and we arrive at the Bowman's capsule. So the glomerular capsule, again also called Bowman's capsule, is formed by an internal visceral layer directly lining the glomerular capillaries, and it's also then formed by an external parietal layer composed of simple squamous epithelium. So together, those structures are going to create a space, a capsular space. And that's what the filtrate is then going to move into. The renal corpuscle consists of a vascular pole. That's where our arterioles are going to attach to the glomerulus, and a tubular pole where the renal tubule originates. And we can see that structure there. So in summary, the blood comes into the glomerulus, and at the site of these capillaries, we have small structures called podocytes that are going to help us filter and move those structures through. And let's take a closer look at some of those filtration membranes at this time. So in this diagram, we're going more in depth into what's happening at these capillaries. And we see on the capillaries, we have specialized structures. We have podocytes on these capillaries, and we have fenestrated endothelia. Fenestrated means it has sort of pores to it. So perhaps you can appreciate how we have a sort of physical filtration, almost a sort of sieve that it's going to require some larger proteins to be not moving through. So this filtration membrane is a porous membrane. It is thin and it is negatively charged, and again we have physical filtration of molecules. So this, as you can see, this endothelium of the glomerulus is fenestrated. So that's going to allow plasma and dissolved substances to be filtered while restricting the passage of large structures. On a closer inspection, we see this porous membrane then restricts the passage of large plasma proteins while allowing smaller structures to pass through this basement membrane. It is noteworthy that if larger proteins do get out of those filtration slits, we have structures called glomerular mesangial cells that will degrade them. So these cells are positioned in a proper area where they have phagocytic properties. That is, they are able to engulf them and break them down. Now we go a little bit more in depth into the renal tubule. That means we're coming out of the renal corpuscle. We see the renal tubule consists of, again, a proximal convoluted tubule, again, because it's closer to the glomerulus, leading to a loop of Henle, aka nephron loop, and then coming into a distal convoluted tubule. Here we go a little bit more in depth into some of the histology of the nephron. Let's go ahead and look at how some of the structures we've discussed so far have specialized cells that assist in their functions. And these are going to be specialized form of epithelia. So we then see that in the Bowman's capsule, we have those podocytes, again, which are special structures for filtration. And also that parietal layer of the capsule has a special type of epithelia. In addition, in the proximal convoluted tubule, we see that we have special cells that have microvilli on them. And those are going to be tiny, quote-unquote, hair-like structures that exist on the surface of the cells, very similar to those which are found in the digestive system. When we move through the descending loop of the proximal convoluted tubule, we arrive at the nephron loop. We're going to have some simple squamous epithelia that lines the thin segments of each limb, and then simple cuboidal epithelia is going to align those thick segments. The region of the renal tubule originating from the thick ascending limb of the nephron loop is lined with small, simple cuboidal epithelium and short apical microvilli, that's going to be part of our distal convoluted tubule cells. And then lastly, we see that in the collecting duct, we have cells called intercalated cells and principal cells. These principal cells have cellular receptors that bind both aldosterone and antidiuretic hormone together. And then in this collecting duct, we also have intercalated cells, and these are going to be specialized epithelial cells that help regulate urine pH and blood pH. So in summary, each part of the nephron has its own unique epithelia that relates to it. So in our discussion of the nephron, we go a little bit more in depth into a structure called the juxtaglomerular apparatus. Thank goodness it's abbreviated by JGA. And this is a region where afferent arteriole makes contact with the ascending limb of the loop of Henle. And that's what we mean by kind of just juxtaglomerular. It's around the glomerulus. And so at this region of the nephron, we're going to have regulation of filtrate, and we're also going to have blood pressure regulated as well. And this is done in two ways. We have juxtaglomerular cells of the afferent arteriole, and these are going to be smooth muscle cells that contract when they're stimulated or stretched by the sympathetic nervous system. Also, we have structures called granular cells that are going to release renin. In addition, we have a structure called macula densa. Macula densa is a group of epithelial cells. These are going to be in the wall of the distal convoluted tubule and also the tubule side of the afferent arteriole. And these are going to detect changes in sodium chloride concentration. That means they're able to pick up the concentration of salts within the lumen of the distal convoluted tubule. So that's going to signal the granular cells to release renin and help maintain a homeostatic balance. So our last slide we discussed in this section just simply mentions the number of nephrons. It's noteworthy that the number of nephrons remains constant from birth. Therefore, any increase in the actual size of the kidney is an actual size of the increase of individual nephrons. If these nephrons are injured, no replacement occurs. Very complex structures of epithelia that cannot be cloned very well. Generally speaking, these nephrons do not become dysfunctional until about 25% of their normal functioning declines. And that means that other nephrons can handle the extra work by picking up the slack, so to speak. Interestingly, if we get one kidney removed, it causes the enlargement of the remaining kidney to compensate for that loss. So in summary, in this first section of the urinary system, we see some major structures of the kidney and some blood flow. Next section, we go more into filtrate and the specifics of how filtration occurs during all those structures we talked about.